or different sample that actually endophytic or the fungi that endophytic. Yeah, so for the biocontrol ones. So in terms of the secondary metabolite, they are very diverse. So, and then this also, yeah, the semitatum, one of the example, the saprophytic, but now we have a few isolate that's actually pathogenic. So we also study on the endophytic one. So the problem is when we eat just now, yeah. So what is actually the problems when we eat the infected one? So this fungi is produced secondary metabolites. Yeah, one of the metabolites that's mycotoxin and can cause a mycotoxin that's harm to us, yeah, humans and also animals. So we need to actually to avoid, yeah, try, yeah, to avoid eat them. So what we did is we purify, yeah, and then we identify, we amplify the gene and also that's present or not, yeah, for the indicator, and then we quantify the production. I just pick one of the toxins here, FP1, yeah, and then the problem is most of our sample, if they are present of the ascomycete, for example, fusarium here, they might produce this toxin. I jump to, because I know I only have 30 minutes, yeah. So when the problem is when they produce, yeah, they be able to give the effect during the development. That's, we should worry. For example, here, the control one, there is no edema, but when we expose them to the small, the little of the toxin, yeah, FB1, so they have the, actually that's a blood clotting, yeah, this. And here you can see, yeah, the zebrafish larvae at 120 hours, yeah, post-fertilized. Yeah, you can see there is the problems, yeah, the abnormal growth, yeah, they have the pericardial edema, yeah, and also they have a yolk sac edema, yeah, compared to the control. So you just imagine we eat for 40 years, yeah, these fruits or any foods that contaminated with ascomycete. Some of them may be from the fusarium. This fusarium produced everyone's, yeah, for today maybe 0.001 microns, yeah, them, yeah. So, but if they are bioaccumulate in our organs. So what happened after 40 years? What happened after 20 years? Yeah, because, yeah, you might say that, yeah, we cook the food before we eat. They will denature. Yeah, it's not 100%. This toxin will denature at 100 degrees Celsius. Yeah, they will bioaccumulate in our body. Yeah, and then they might, yeah, because of from the foods, they might activate, yeah, some of the cell cancer, or they might is actually the factor that actually start, yeah, the organ failure. Okay, so, and then, okay, this one, yeah, I know that's in Andalas as well. Many researchers that focus on the trichoderma species as a biocontrol of the plants. We also did this one, yeah, and as you agree as well, yeah, we need a biocontrol, yeah, for the, yes, they are expensive compared to the chemical one, but is it safe, the chemical? Yeah, because of the growers, the farmers, yeah, it's just very easy to apply, and the pest will kill, the pest will die, yeah, but they are broad spectrum. It's not only the pathogenic will be there, but the good one as well. So that's the reason we need, yeah, to minimize, yeah, use of the chemical pesticide. Sometimes we have no choice. yeah, we have no power, yeah, to control the use of this chemical pesticide. But the awareness, yeah, that's very important for us to share, yeah. So the awareness, at least we did something, yeah. So, and this mode of actions, of course, yeah, for either they are direct mechanism, yeah, by producing, yeah, antibiotic, yeah, for the antibiosis, yeah, parasitism, or indirect mechanism, competitions for the nutrients, for the space, yeah, and also induce the systematic resistance. Actually, there is too many others, the mechanisms, yeah. So, and then you can actually apply it, yeah. So the biocontrol is not it actually is not new in our country, yeah, in Malaysia or Indonesia. We have many products, yeah, that's with the biocontrol. But the problem is the survival rate of this trichoderma, yeah. So that's the reason we study on the, yeah, this only example that's this is banana disease, the wilt disease. Just now is Fusarium earroot, yeah. So actually they give a good effect. And we also look at on the growth and photosynthetic performance, yeah, Trichoderma aspergillum on the Fusarium solani as well. So, and you can see here, they have a group performance, a growth performance compared to the control. And like I said just now, the problem is actually the survival rate of the trichoderma. So this for the culture filtrate bioassay. Because of that one, we use a few agriculture waste as a carrier. Yeah. So we try to enhance this colony, yeah, to increase the colony of the trichoderma. So before we apply in the field. So the coconut fiber and also empty fruit branch is actually one of the medium as a carrier. Actually, you can use to culture this trichoderma. And you can actually freeze dry, yeah, the inoculum, yeah, the conidia suspension of this trichoderma. And then you can actually inoculate on the carrier. Okay, so, and then we also study these two, another isolate, yeah, and give also the oil palm husk and also the coconut, yeah, as a best medium. So this another study, yeah, that we use for oil palm plantations, yeah, versus stem rot. Okay, so another is that I want to share, yeah, about the use of the plant extract as a biofungicide against the post-harvest fruit rot pathogens. Yeah, we have many actually because of our tropical country. We have many plants actually we can when try. Yeah, the best is actually look at on the food or the agriculture waste. So what we did, yeah, so because of, you know, that about the 42%, yeah, the post-harvest loss, yeah, in Southeast Asia, and 20% is actually post-harvest loss in Malaysia. So the major is, yeah, we know, yeah, most of them is actually from the ascomycete, yeah, other than that, the other microbes lah. So, and the common practice is use fungicide. So, but we know fungicide is harmful to us, yeah. So we try, yeah, to have the alternative. And of course, for the application, we need to more creative, yeah. So actually, it's not only the plants extract, we have the essential oil, yeah, that's working on this one, works on this one. So this is the analysis, yeah, that we study. We use the a few plants, yeah, that's the best that we found out is actually the Gracina mangostina, the pericarps of the mangosteen. So it's not only these plants, actually we have many few plants that we tested. Yeah, so we did on the in vivo, in vitro, and also the in vivo, and we did also the fungicidal activity test, yeah, the germinations, and maybe the most interesting that I want to share, yeah, because of the limited times, yeah, about the morphological changes, yeah, observations, yeah, for the SEM and also TEM. Yeah, based on these observations, yeah, for the usually, yeah, this pathogen, yeah, produce the Fusarium oxysporum on the SNA, yeah, produce the crystal form, but they have inability, yeah, they lost the ability to produce the crystal form, yeah, when we treated with the mangosteen pericarp. And then also the abnormal morphological characteristic, yeah, when we observe under SEM. So, and then when we cross this hyphae, yeah, the cell that showed the abnormal shell shape, yeah, that's actually all this abnormal shape, yeah, bring the cavities, yeah, the vesicle. So, yeah, this is actually the gift effect to the growth, and it's actually the good ones, yeah. But we need to check, yeah, the toxicity level of these extracts. Yeah, actually that's a in future we actually plan to do it. So the normal cell list you can see, yeah, the from the top left, yeah, this actually the normal one, yeah, for the infected, yeah, of the treated. Yeah, even for the carbendazime as well. Yeah, for the treated, yeah, the secretions of the cell wall, cell membrane, and also the cytoplasm. You can see this one. So not only for use as mangosteen, yeah, we also use the Alesium sativum, yeah. So, and you can see here it's actually very good, yeah. Actually, they reduce, yeah, and inhibit and almost kill, yeah, the pathogens. So the latest, yeah, we use the pepper, yeah, sarmentosum, yeah, in Malaysia we call a daun kadok. I'm not sure in Indonesia what actually you call these plants, yeah, the common name. So actually they have a good, yeah, actually for the crude extract, yeah, we apply. So, and when we compare to carbendazime, yeah, the positive control, the commercial fungicide, actually they can actually help, yeah, to control and actually inhibit the growth of the plants pathogens, yeah, these fungal pathogens. So the yes, when you study on the plant disease, plant pathogens, plant protections, yeah, there is too many methods, yeah, to control, but how actually effectively we want to apply in the field. Yeah, that's actually a long way to go. Yeah, we hope that's we can actually help, yeah, for this control the disease. For the take home message, yeah. So I hope that we are be a part of the solutions, yeah, it's not the pollutions. So I end my sharings, yeah, acknowledge to all my former students, yeah, and also the university and the agency that's contribute to these compilations of the studies. So I'm welcome for the question or suggestion or any opinions, yeah, or the collaborations, yeah, in future. Yeah, I know that's the time's up already 13 minutes. Yep, dah melebihi, right? 30 minutes. With that, yeah, you can scan my profile and we can have further discussions later if needed. Okay, thank you. I pass to Miss Rosiana. Yeah, for moderator. Thank you again.
Okay, thank you very much, Ibu Nur Ain, for nice presentation. And I already resume some your presentation, Bu. And as I communicate, include fusarium. We find fusarium if everywhere, and we use biocontrol to good effect to control fusarium, and we have potential biofungicide of control fungal disease. So thank you very much, Ibu Nur Ain, and I hope you can still with us until the end the session. And for the next, for all the participants, if you can have a question now, and you can write your question in the chat box, and I can help to write letters. So the second lecture, we invite Professor Ahmadi Priyatmojo from Gajah Mada University. Are you hear me, Professor?
Okay, thank you, Ms. Rosiana. Can you hear me? Can you hear me?
Okay.
Yeah, clear.
Clear. Thank you.
Okay. Let me tell you about his curriculum vitae from Professor Ahmadi Priyatmojo. He is a lecturer in Universitas Gajah Mada and education bachelor degree Faculty of Agriculture in Universitas Gajah Mada, and master degree plant pathology Gifu Universitas Japan, and PhD program in plant pathology Gifu University of Japan. And expertise of Professor Ahmadi biological science and plant pathology. And achievement published 45 journal papers, and he got any awards from Professor Ahmadi Priyatmojo. So Professor Ahmadi Priyatmojo will give lecture about next, please.
Before.
Okay. Professor Ahmadi give lecture about current status of seed rot disease on rice caused by Fusarium. Professor Ahmadi, you have 30 minutes for presentation. This time is yours.
Okay, thank you, Ms. Rosiana, moderator. Okay, first of all, I would like to gratefully thank you for inviting me to this webinar. I would like to greeting Dean of the Faculty of Agriculture, Dr. Indra Dwipa, and then Head of the Department, Professor Dr. Nofri Nelly, and also Associate Professor Nur Aini Sati. Assalamualaikum warahmatullahi wabarakatuh. Okay, I would like to deliver my presentation entitled, sorry. current status of set rot disease on rice caused by Fusarium species. I just focus on this title, especially on the rice, and the causal pathogen is Fusarium. As mentioned before, that Associate Professor Nuraini Isati show us the global, the escomicity in the words, and in this second session, I would like to deliver this title. As we know that symptom of the cetrot on rice we can found in the set, and then the develop of the disease, we can find in the panicle. Yeah. So after that, we cannot harvest the rice because all of the part of this season, sorry, all of the part of this panicle and the set is roots, and in the susceptible resistant, they can attack almost all the rice and will destroy the rice, and the losses is about more than 80%. As we know that the pathogen of the set rot on rice caused by many pathogens, such as Fusarium species and Salcladium oryzae. Another pathogen caused by the bacteria Pseudomonas fuscovaginae. This is the three major pathogen of the set rot on rice. Recently, our PhD student, found the other species of the bacteria, Xanthomonas saccari, isolated from the rice set rot disease in Lampung. So, okay. Cannot move. It cannot move. Sorry, I will start again. Please wait a moment.
Ada yang bawa headset? Ada yang punya headset? Nggak, lo terdengar.
Okay. Sorry. Yeah. As we mentioned before, that there are many pathogens on the set rot on rice, and the symptom is similar. We cannot differentiate them by just morphology of the symptom. But some of the pathogen, for example, Fusarium subspecies, is produce sign of the disease, the colony of the mycelium, in the high relative humidity condition. but for the Gladium, Oryzae, and Cydonias, we cannot find any signs of a sign of the disease. So it's very difficult to know directly from the symptom of this disease in the field, and then to know the species of the pathogen of the seed rot, we have to go to the laboratory and do isolation in order to know the causal agent of this disease. My student isolated from the different center of padi field in Indonesia, from the North Sumatra till East Nusa Tenggara and some part of the Sulawesi, especially in the South Sulawesi, in the center of padi rice production, the rice production. And then, sorry. And then I just focus on the Fusarium, Fusarium species, the members of the Ascomycete here. So this is the part of the dissertation of my student. From the location in the Indonesia, from North Sumatra to East South Sulawesi, some of the species of the Fusarium belonging to the Fusarium incarnatum equisity species complex. Yeah. And then others belonging to the Fusarium fujikuroi species complex. Other Fusarium, other here is just two species, Fusarium solani and Fusarium oxysporum. So from the one of the single symptom, and then from the different location, we can find, we can find a complex of the species of the Fusarium here. So, for example, in the North Sumatra here, we have almost similar numbers of the species of the Fusarium for the Fusarium incarnatum equidity species complex, and also for the Fusarium fujikuroi species complex. But if you look here from the West Sumatra, from the one field, only we can find Fusarium incarnatum equidity species complex. None of Fusarium fujikuroi complex found here. On the other hand, in the Bengkulu, we found Fusarium fujikuroi complex, but now Fusarium. Sorry, that is. Okay, thank you. None of the Fusarium incarnatum species complex found it in this region in Bengkulu province. A similar result also in the other location here. So, for example, here East Java, Jawa Timur, East Java, we found many of Fusarium incarnatum as equidity complex, but we could not found the Fusarium fujikuroi species complex here. Yeah, this is the pathogenicity test for the Fusarium isolate that we found it, and almost all of the Fusarium could in the set of the rice here. Yeah, you can see the symptom here. similar to the symptom that we can find in the field. We make it by the artificial inoculation. And some of the isolate produced, you can see here the sign of the disease, Miselia. Of this is also there's a Miselia here. So from this result, we concluded that all of the fusarium that we collected from the different region in Indonesia could infect the rice. This is some of the morphological characteristics of the fusarium that Professor Isati known very well about this morphological characteristic of this fusarium. Yeah, some of them produce microgonidia here, and there is diverse in the culture color. This is the isolate from the Fujikuroi species. Here, the color is violets. Some of them produce microgonidia. This is also belonging to the Fujikuroi complex, species complex. Yeah. Fujikuroi species complex here. This is macrogonidia. This microgonidia and microgonidia. We can easily found in the Fujikuroi complex here. And this is the isolate from the other Fusarium solani. Yeah, we can microgonidia. So because of the morphological diversity of this Fusarium, it's very difficult to identify them for the species level. Therefore, the molecular identification is needed, to identify in the species level of the Fusarium. This is just a method of them, and we use some primers of to identify this. Yeah, this is a common primer for identify the Fusarium. Yeah. Similar with the Professor Isati mentioned before. Yeah, we have experience with the Suga Sensei in Japan. He is also expert in the Fusarium species. So from the isolation from the many locations in Indonesia, we found some of the species here. For example, this is by the molecular analysis. For example, of Fusarium Fujikuroi species complex, we could find Fusarium cedohirginatum, Fusarium sakari, Fusarium andiyasi, Fusarium fujikuroi, Fusarium proliferatum. And from the Fusarium incarnatum equisiti species complex, we could find Fusarium puparinum, Fusarium hainanensis, Fusarium sulawesiensis, and Fusarium tanabumbuensis. Other species is a Fusarium oxysporum species complex and Fusarium solana complex, but there is not so many of this isolate we can found. The two major Fusarium that we can found is a Fusarium incarnatum equisetis species complex and Fusarium chicoroi species complex. And then how to manage the cetera on the rice here. Yeah, we try to inoculate some of the cultivar that commonly cultivated by the farmer in Indonesia. For example, Ciherang variety, Situ Bakendit, Mekonga, Inpari 30, Inpari 32. Yeah, with the some isolate of the Fusarium here, and for example, here is Fusarium hainensis. Below here is Fusarium pubalinum. This is a Fusarium sulawesiensis. And some of the cultivar shown the different reaction by this artificial inoculation. Yeah, for example, in the Situ Bakendit, yeah, susceptible here, Bakendit. More moderate susceptible is in the Mekonga, in Pari, and in Pari 32. And for the moderate resistant is in Pari 30. So by different isolate, maybe some different reaction for the artificial inoculation on the cultivar we can find here. But yeah, but we can see here almost of the cultivar that only used by this farmer in Indonesia could infect by Fusarium species. Yeah. This is a Fusarium proliferatum. Here is Fusarium pseudokirchhanatum. This is a Fusarium Buchikuroi complex. Yeah. So by this result, we can not find a resistant variety. Yeah, it's difficult to find resistant rice variety to the center of disease. That farmer usually want to plant rice with high productivity. Yeah. So maybe in the future, the resistant variety is needed, yeah, to solve problem for seed rot disease in Indonesia. Maybe this is need more time, yeah, to develop the resistant variety on the rice, especially to the seed rot disease. And then how to know the initial inoculation, yeah, in the field by the different pathogen of the seed rot disease, as I mentioned before, that the three major pathogen are associated with the seed rot on the rice: Sarocladium oryzae, Fusarium, and bacteria. So here we try to isolate the pathogen on the different days of the planting here, yeah, 62, 76, and 90 days. So compare to the Sarocladium oryzae, Fusarium species could isolated in the high frequency on the 19 days. Yeah. But in earlier plantation, we found actually Sarucladium oryzae here. So maybe in the recommendation of the management of set rot disease, so we can notice here that Sarucladium oryzae usually infected the plant in the early, yeah, compared to the Fusarium species. Yeah, because in the field it's very difficult. We identify which one is Fusarium, which one is Sarucladium, just from the symptom itself. It is very difficult. And then my student studied the isolation frequency and found that in the early plantation, we found Sarucladium oryzae in the high isolation frequency, higher isolation frequency than Fusarium. And until now in Indonesia, there is no recommendation of the chemical pesticide used for the set rots. So my student studied some of the pesticide here, yeah, to control the set rot disease, and she found that some of the pesticide here can control set rot disease. For example, the hexaconazole, carbendazim, and mancozeb. This is the control without the fungicide. Yeah. Sorry. So this is still a difficulty to control of the set rot disease, yeah, based on the symptom in the field. Sometimes the farmer didn't know the diseases because it is very similar to the set bite caused by Rhizoctonia solani, and also sometimes similar to the rice blast, yeah, and the panicle. And then other disease that similar to the set rot is the BG on the grain of the rice. Yeah. But the identification of this pathogen is very needed to know the recommendation of the management to this pathogen. Yeah, because if we cannot know the pathogen, we can do the recommendation for controlling this disease. But, this is all of my presentation. Thank you very much. Okay. I would like to receive the question or suggestion from all of the participants here. Thank you very much.
Okay, thank you very much, Prof. For the next presentation, and I got some how to manage the set rot disease on rice cows, use resistant varieties. And then farmer can hike production. So, Bapak Ibu, ladies and gentlemen, we have time for discussion. For all the participants, if you have a question, you can ask a question by raising your hand, and you can write your question in the chat box. So I will try to read the chat box. So in the chat box, I see question for Pak Priyatmojo.
Okay.
For Prof. Priyatmojo, a question is, it's known that many rice plant disease cause significant loss but don't cause crop failure. Why is that? And is it possible then that in the future, the main pathogen of rice plant will cause crop failure? To Professor Priyatmojo, you first answer you have from Professor Ahmadi Priyatmojo. Please, Prof.
Sorry, Rosiana.
Oh, yeah.
Which one the question? I just can see the from my Shahrawati.
In the chat box.
Mm-hmm.
Oh, sorry, Prof. Okay.
Okay, please.
From first from Bu Maisarawati.
Okay.
Could you explain more what kind of biofungicide that apply to control set rot in Indonesia?
Okay, thank you, Bu my Sarwati. Yeah, until now we don't have a research about this biofungicide to control the set rot, but some of the another researcher said that some of the fungi, for example, Trichoderma, and other is Bacillus, can use to control this set rot disease. Yeah, but my team now didn't have this research specific in the term of biofungicide. Sorry.
Okay. This is answer from Professor Priyatmojo. And I have question for you, Prof.
Okay.
In Indonesia, how the development of Fusarium fujikuroi now, Prof, in Indonesia, how the development?
Okay. As mentioned before that from I will say first. Sorry. This one. This one. Yeah.
Okay.
Okay, you can see it. Okay. From disease symptom, we can easily isolate Fusarium fujikuroi species complex. Yeah, here my student Safika Pramuna Dipta found some species of Fusarium pseudohircinatum, Fusarium sakari, Fusarium adiyasi, Fusarium fujikuroi isep, and Fusarium proliferatum. Some of these species, yeah, recently can infect not only the rice but some, for example, Fusarium sakari, of course, in the what you call sugar cane. Yeah. And as you know, for the Fusarium proliferatum, there are many reports could infect many kind of the species of the plant. So maybe we have to aware about this Fusarium fujikuroi species complex. Thank you.
Okay. Thank you very much for your answer, Professor. The next question from Professor Novrinelli for a question for me, Ibu Nurain Izati. The question is, has Fusarium also been used as an entomopathogen?
Yeah, actually, based on the previous report, yeah, some of the species, Fusarium, for example, the Fusarium incernatum equistae, that's actually be able, yeah, as an entomopathogen. Yeah, so yes, and also some, I couldn't remember what is the species, but actually one of the species of Fusarium incernatum equistae. Before this, we have a different taxonomies. Yeah, Fusarium equistae is one of the species. Yeah, but now they have a species complex. One of the common one that have the entomopathogenic, yeah, entomopathogens. So that's a Fusarium incernatum equistae. That's example. Yeah, you can actually use that one. Try to look at is if this non-pathogenic to plants, and you can actually try to look at and is it they are have the efficacy, yeah, effective to control the this pest. Okay, thank you. Is it I'm answering the questions? Answer the questions?
Okay, thank you very much, Ibu Nur Ain. And the question from Hasan, the question for Professor Piatmojo. Question is, from every isolate fusarium that you have been isolate in every area in Indonesia, which fusarium do you concern that has a high level of virulence, especially for seed root disease?
Okay, thank you. Ms. Luciana. Thank you
Professor.
for Pak Hasan Alapamat. That yeah, as far as I know, that's all isolate the fusarium that we collected from the many different locations in Indonesia is virulency on rice, yeah. Not only from Fusarium incarnatum equisiti complex or and Fusarium fujicore complex that we know some of the, this complex has many species. So almost of the isolate could affect the rise in the high level of the virulence. So I cannot mention one of the isolate here. Thank you.
Okay, thank you very much, Professor, for your answer. And the next question from Ibu Masyarawati. This question is, ascormicate can produce toxin and impact on human and animals. Could you explain more, Ibu Nur Ain Izati, please?
Okay, so as we know that ascomycete produce these secondary metabolites, that actually one of the metabolite is toxin and cause a mycotoxin to humans and also to animals. So for example, I just give example, Fusarium will produce that fumonisin, monoliformin. So this can actually have the effect on the kidney failure and also neurological disease. Yeah, that's and also can cause for example that leukoencephalomyelitis Malaysia. So that's the problems about the toxicity that's caused by FB1, for example. So and then this not only, yeah, if we eat or we inhale, yeah, so they can give effect to us. And if let's say that this animal, for example, yeah, you give chickens, yeah, with a corn meal that infected with fusarium, then this fusarium is be able to produce FB1, for example. So the chicken will contaminated, yeah, have the toxin, and we eat this infected chickens. So actually they can bioaccumulate in our body as well. So the internal bleeding also is one of the effects, for this toxin. Actually, there is too many disease that actually relate with this toxin. So, and most of usually that the common one, that's internal bleeding, the organ failure, for example, the kidney failure. So it's not only for short term. Sometimes they have the effect on the long terms. If you eat in small portions, that's a bioaccumulates. So that's actually an example of the effects of the toxins, to humans and animals. Okay, thank you.
Thank you very much, Ibu Nur Ain. I have question for you, Ibu Nur Ain Izati. In Malaysia, how about potential yes epiphyt control the fusarium? Bu, can you explain?
Yeah, that's actually very good question. So actually you can, that's a potential. So but you have to make sure that you make sure that you undergo all this, the in vitro and also in vivo, then before you apply to the field. So actually there is the potential for that one.
Udah.
Okay, thank you very much for your answer, Ibu Nur Ain. There is any question
Can I add
from
some
all
information,
participants?
Rosiana? Can I add some information here?
Oh yeah.
Okay, thank you. Yeah,
Okay.
some of the isolate that we isolated from is also mycotoxin, and the paper is published in the IUP conference, so you can find it out there. Thank you.
Okay, thank you very much, Prof. and Ibu Nur Ain Izati from Malaysia. In the chat box, I don't see the question, and I think this is the end of the session. Ladies and gentlemen, this is the end of the session. Thank you very much for both speakers today that already share knowledge and her experience for us. And I hope we can continue to work together in the future. And I hope to see you again. And to all participants, thank you very much. I hope see you again. And I back to Ira.
Thank you very much to our moderator, Rosiana Fadilah, and speakers today, Associate Professor Nur Ain Izati and Professor Ahmad Priyatmojo. Ladies and gentlemen, moving the next agenda, closing and appreciation. The certificate will be given by the head of Plant Protection Department, Professor Nofrinelli. To Professor Nofrinelli, please, time is yours.
Okay, thank you, Ira. Pak Prof. Ipi, Prof. Izati, thank you very much for your sharing knowledge. And now we give you certificate. I hope you can accept this certificate, and thank you from Andalas University. This is for Pak Ipi.
Thank you very much, Prof. Nopri. Thank you very much.
Thank you. And next for Bu Izati, I hope you can accept this certificate.
Recording in progress.
Thank you for sharing your knowledge. I hope the next weekend the collaboration, maybe not only publication but maybe research. Pak Ipi from UGM, Bu Izati from UPM. I hope we can the collaboration for the next research. Thank you very much.
So,
Thank you.
Thank you, Ira.
Thank you very much, Dr. Nofrinelli, Professor Nofrinelli. Okay, ladies and gentlemen, we have come to the end of the agenda. Well, before the end of this webinar, allow me to take a picture of audience. I will guide our photo session today. The audience, please open the camera and follow my instruction. The first slide. 1, 2, 3. Okay. The second slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. The fifth slide. 1, 2, 3. The sixth slide. 1, 2, 3.
How many slides?
10 slide.
Good.
7 slide. 1, 2, 3. 8 slide. 1, 2, 3. And the last slide. 1, 2, 3. Okay, thank you very much.
thank you.
Thank you, Bu Nur.
Okay. Well, ladies and gentlemen, I hope you enjoy and benefit from the presentation and discussion of our distinguished speakers. I would like to thank them once again for their valuable contribution and for making this webinar a success. I hope to see you again in the next webinar. Thank you very much for your attendance and your nice attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Makasih Pak Ipi,
Iya,
Bu Izati.
Iya, Bu
Terima kasih, Bu.
Nurain.
Ya.
Makasih ya Pak Ipi.
Pamit. Assalamualaikum.
Silakan di
Iya,
Pamit ya.
kita tutup.
Recording stopped.